Buenos días. Antes de comenzar con nuestro simposio, que hemos llamado Chagas Disease, Immunology and Immunotherapies Virtual Symposium, las organizadoras Ana Rosa, Karina y yo, Andrés, queríamos darle la bienvenida a todos los oradores y la audiencia y agradecerles su presencia en esta jornada. También queremos agradecer a los auspiciantes que han posibilitado la concreción de esta serie de charlas científicas. Ellos son el Consejo Nacional de Investigaciones Científicas y Técnicas de Argentina, o CONICET, la Facultad de Ciencias Médicas de la Universidad Nacional de Rosario, la Sociedad Argentina de Protozoología y Enfermedades Parasitarias, el Instituto de Investigaciones en Ingeniería Genética y Biología Molecular, o INGEBI, del CONICET, la Sociedad Argentina de Inmunología, el Instituto de Inmunología Clínica y Experimental de Rosario, o IDICER, también del CONICET, y finalmente el Ministerio de Producción, Ciencia y Tecnología de la provincia de Santa Fe. El simposio entonces consistirá en 3 reuniones, los lunes 8, 15 y 22 de marzo, de 10 a 13:30 en el uso horario de Buenos Aires. And now I will switch to English and welcome our non-Spanish speakers. We would like to thank you all and to express our gratitude for your support. It was very easy to organize this symposium with your help. This is our very first symposium and hopefully not the last one. Taking profit of virtuality, we will try to keep this communication channel open as much as we can. Regarding our program, you can see here our speakers today, and I will inform you that all the speakers will have 30 minutes for their talks, and then the audience will be able to ask questions for 10-15 minutes. But we will not open the microphones to avoid confusion, and hence, we gently request you to write your questions in the chat, and we will read them after the talks. We also ask you to keep your microphone silent for respect. If time was not enough for your questions to be answered, you may write us to our email and we will forward them to the speakers and go back to you afterwards. Now, I will introduce you our first speaker, Professor Rick Tarleton. Dr. Tarleton is a Regents Professor at the University of Georgia in the Biological Sciences Center for Tropical and Emerging Global Diseases from the Department of Cellular Biology. Dr. Tarleton has published more than 150 manuscripts and is one of the major contributors to the understanding of the immune responses elicited during Trypanosoma cruzi infection. He has developed many interesting hypotheses regarding how Trypanosoma cruzi evades the host response to develop persistent infections, from insufficient TLR activation to dormant-like parasite resistant to almost everything. Today, he will tell us a story about this, named Trypanosoma cruzi, Mechanisms of Resistance to Immunological and Pharmacological Assaults. Thank you again, Rick, for joining us today, and please go ahead with your presentation.
People in the audience that the lack of, or the presence of persistence is not due to the lack of strong immune responses or to an overregulated immune system. I think actually the immune system sees T. cruzi very nicely and controls it very nicely. But T. cruzi does do a number of things that I think are really important in its success. One is that when it invades host cells, it does so in a very stealth manner and very weakly triggers sensors that are necessary to initiate immune, especially innate immune responses. And we think this is really critical, not just early in infection, but throughout the infection. We also know that T. cruzi primarily persists in tissues like muscle that have a relatively low level of expression of class 1 MHC. So we deal mostly with immune responses at the infected cell level because this is where the parasite spends most of its time in an infection inside cells. And of course, those cytoplasmic parasites are recognized by CD8 positive T cells that see class 1 MHC associated parasite peptides. But if those cells happen to be cells like muscle, they don't have as much MHC as some other cells. And this means that the triggering of CD8 T cells will be less efficient. And I'll show you a little bit of data for that. We also know that the major targets of the CD8 T cell responses are highly variant antigens, including those from the transalidase family, and that these transalidase variants are being expressed simultaneously. So here we show a single peptide on the surface of cells, but in fact, there are different peptides that are being expressed here coming from different transalidase and other large gene family proteins that are being expressed simultaneously in the parasite. And so what the immune system is seeing is a plethora of different epitopes. And we believe that this may affect how efficiently that CD8 T cell response can work. And so I'll show you a little bit of data on all these things. And this is one of my favorite studies, not done in my lab, but done by Barbara Burley's lab and John Boothroyd's lab, looking at this question of what happens when T. cruzi invades a host cell. And this is work done really looking, I think it was microarray in the early 2000s when this was originally done, but the same sort of data can be collected by RNA-seq these days, showing that when T. cruzi invades, there's almost nothing that happens within the first 6 hours in terms of changes in host gene expression. And it's not until 24 hours that one starts to see some genes start to be up or down regulated. And most of these are interferon response genes, type 1 interferon response genes that are responding presumably to a small amount of type 1 interferon that's being produced from the infected cell. And if you compare that to essentially the exact same system, same host cells, same microarray analysis, but different parasite, this being Toxoplasma gondii, you see a very different picture that within a couple of hours after invasion, a number of genes are being upregulated and downregulated, and by 24 hours, there's a huge change in host gene transcription. So this is one of the issues that we use to argue for the fact that when T. cruzi invades host cells, it's really not being sensed in a strong way. And this doesn't mean that immune responses aren't generated, but it does mean, I think, that the immune system has difficulty seeing very early on that an infection is occurring. And in fact, some of the work in our lab by Natasha is showing that in fact, that early type 1 interferon that's being made actually promotes parasite growth at the site of infection. That if you prevent that type 1 interferon, we can do this with sting knockouts. We know this is a sting dependent response, or using IFNAR knockout mice, which lack the type 1 interferon receptor, that they have a higher first peak of response, but they don't show this a second peak of parasite replication that one sees in wild type mice. Instead, you see a more rapid decline. So the production and the sensing of that type 1 interferon coming from a host cell actually appears to be promoting parasite growth, not regulating parasite growth. So this is I think an important issue to consider in how the immune system recognizes the parasite and starts to deal with it at a very early point in the infection. And it's not just at that early point in infection. We believe this is happening throughout the infection, that every time the parasite goes to a new infection site, there is this delay in the sensing of parasites at that site. And this is happening even in immune animals. And the sort of stand-ins for those sorts of data, we're hoping to do this in a little different way, but the way we have been able to do it so far is to demonstrate that one can reinfect chronically infected mice, which is shown here. So this is again using that footpad model, reinfecting. If you infect naive mice, you get a huge increase in parasite numbers. In this case, we're infecting with a high number of parasites. And the point is to show that you can take even a chronically infected mouse and establish parasites at that infection site, even though it has an existing immune response. So this delay in control of the parasite load until 6-7 days after infection allows reestablishment of parasites. And we think this allows reestablishments or establishment of parasites in various tissues throughout the body, not just when we're re-challenging here. And this is work by Juan Bustamante. This is Angel Badija's work here, showing that one can not just reinfect chronically infected mice, but you can drug cure mice and reinfect them. In this case, he's done it 3 times. Infect mice, drug cure them. Here we're looking at T cell CD127 expression as a measure of long-term memory T cells. And as you cure the infection, you get an increase in memory population. But as you re-challenge these guys, the T cells are now seeing antigen again, and they decrease the expression of CD127. So you can monitor the reinfection and cure. So basically, one can infect, cure with benznidazole, reinfect, cure with benznidazole, reinfect, cure with benznidazole, and reinfect again. So we can go through 3 or 4 rounds of infection and cure, and these animals are still susceptible to being reinfected. So in our view, this is a real challenge to dealing with making a vaccine. If you can't generate immune responses that will prevent reinfection by either having a persistent infection or by having a drug-cured infection, it's difficult to see how one's going to do that with a vaccine. The deal with T. cruzi in muscle has been one that's interested us for a long time. And Angela Pack, who was a graduate student in the lab, worked out this system where we asked the question, if you enhance class 1 MHC expression specifically in muscle, this is in skeletal muscle, not cardiac muscle. She made a mouse crossing a couple of different existing strains where one could use tetracycline or doxycycline to enhance class 1 MHC expression specifically in muscle, and you can see that here. It's not a massive enhancement. It more or less doubles the expression level, but that has a dramatic effect on the ability of not only the generation of immune responses, but the control of infection in these animals. And one can see this across, again, here we're using a luminescence assay to monitor parasites throughout the animal. And you can see quite a dramatic decrease in infection throughout the animal, not just in muscle, but in all tissues when one just enhances by twofold the amount of class 1 MHC being expressed. So there does seem to be some advantage for being in tissues where there's low MHC. You increase that MHC and you take away that advantage. The problem with this system, this was inducing for 20 days. If one induces longer than that, one sees the reverse situation happen, that actually you now exhaust the immune response and you go from having a ultimately controlled infection at 30 or 40 days to one that's wildly out of control. And this is a clear T cell exhaustion phenomenon, because we can inhibit it with by blocking PD-1, PD-L1 interactions. And so this is the only case actually where we've ever seen a real regulated immune response occurring. So when we enhance the amount of antigen being seen by the immune system by this MHC overexpression, we actually exhaust T cells. And this is a rapid phenomenon that we haven't ever seen. One, we do observe some sort of exhaustion, especially in human subjects who've been infected for many, many years. But in the mouse system, in our hands, this is a very difficult thing to do. And here we see it. So So there is some advantage to having higher MHC expression, at least initially, but eventually this as a disadvantage in that the immune system gets exhausted much more rapidly. And then this issue of T cell recognition of parasites in tissues, we've started to address using light sheet microscopy. So some of you may be familiar with this. We've published a little bit of data on just looking at parasites in tissues where we can take heart or skeletal muscle and clarify it, and then use light sheet microscopy to be able to look at sheets of light and then reconstruct those sheets into a whole tissue. And this is skeletal muscle here. The red are parasite infected cells, and the blue are T cells that have come into the area. And what I want you to notice is the distance between the T cells and the infected host cells, virtually in all cases. And so although you see this inflammation into the tissue, you don't see those T cells really coalescing around these infected cells. And we think this is part of this lack of recognition that the immune system has to say, okay, that cell is infected. Yes, there are infected cells in this tissue, and that's why there's inflammation, but actually identifying those infected cells early in the process, that doesn't happen terribly efficiently. So the third part of this, so I've talked a little bit about the early response and the role of MHC and antigen presentation, but the targets of this response also is one of the factors that I think prevents the T cell response from being more efficient than it could be. And so this is a Centeny plot of two strains of T. cruzi, Brazil, clones from strains, the Brazil strain and the Y strain that we've recently published the sequence up from. We were able to get very nice long read sequence, and using Hi-C in Chicago also to really bring a high quality genome, contiguous genome together. And you can see that this is basically just plotting where there's synteny between chromosomes in the Brazil strain on the right and the Y strain on the left. And what I really want to point out is the gaps where you don't see synteny. And that is in areas like here and here, where the chromosomes are made up, in this case, almost totally of members of these large gene families, the transalidases, the MAS, mucins, and GP63. And so two of the largest chromosomes in Y in Brazil are almost totally made up of these large gene families. And you don't see synteny between these, not because they don't have the same genes, but because the genes, the transalidase, MAS, mucin, and GP63 genes they have in these strains are different from each other. And if you do a best match comparison between the transalidase genes in T. cruzi versus, in Brazil strain versus Y strain, or Y strain versus Brazil strain, you basically find none of those genes that are exactly the same in those two strains, that they're all different from each other. And we also know from looking at the process of how these differences are generated that there is duplication, dispersion, diversification through mutation and through recombination that maintains this repertoire of antigens in these gene families that are different from each other. And there's a lot more data that goes along with this, but I don't have time to talk about it today, but we believe this is a really critical part in a sort of a different way of doing antigenic variation rather than a classical way of making antigen variants and expressing them one at a time. T. cruzi expresses many of them at the same time. And this is appropriate, I think, for a cytoplasmic parasite that's evading a largely CD8 T cell mediated immune response. And so we think that this antigenic variation is an important evasion mechanism because it allows expression of many different protein variants, and these are evolving over time. And the composition of these families is high variation, and they're unique to each isolate, and new variants are being generated all the time. And interestingly, this mechanism for allowing all this variation actually creates some interesting problems for T. cruzi that doesn't exist in some of the other kinetoplastids especially. And so we've been interested in how T. cruzi deals with those problems. These are non-immunological issues, but I think they're important in terms of the biology of the system. And so some of the questions that we're interested in dealing with in the future is how rapidly these new antigen variants are being formed, and are they, do you see them occurring more rapidly or more abundantly when immune pressure is altered? We have some data already that when you infect an animal with a clone of T. cruzi, that population becomes non-clonal fairly quickly. And what the consequences of deletion of large numbers of these antigen variants might be on parasite persistence. So if you take away the ability to make, or the parasite to make all these variants, does that really affect the immune response? And this is something we've been working on for several years. We've had some success and a lot of failures in this part of the project. As you can imagine, it's not an easy thing to do. Or if you could inhibit the generation of new variants, could you resolve the infection more rapidly? And I think the bigger overall question here is, can the basic biology of this parasite host infection with the combined immune evasion mechanisms of a silent entry into host cells, persistence in tissue that have low MHC, and this constant generation of antigen variants, can this be overcome by vaccination or other sorts of immune enhancers? And my personal view is I have a lot of doubts about that possibility, but I'm sure it'll be a point of question and discussion. Very quickly then, I just want to emphasize the drug side of things to say, in our mind, this is an area that's ripe for development. And the issue here, I showed you some of this data already. And the issue here is that there are parasites that are not susceptible to being killed. And one can see this in vitro. You can treat cultures for over 30 days with drug and take off the drug, and there are still parasites that are able to grow out of those cultures, and those parasites are not drug resistant. They have the similar susceptibility to drug as the starting population. They just are not susceptible at that point in time. And we know this is related to this issue of dormancy. And so here we've done these experiments where we cell trace violet stain parasites. And as the parasites develop in host cells, we stain them as trypanomastogotes, infect to generate amastogotes in host cells. They dilute the cell trace violet, and so you only see their expression of the marker protein TD tomato. And here, this is a single infected cell that has a single dormant parasite in it here. Everybody else is dividing very nicely, replicating, but the dormant parasite is not replicating. So what is it about this dormancy that, first, what's the mechanism, but also is this the cause of the failure of drugs? And we think this is a large part of that. One can see these dormant parasites in vivo, and one can treat for long periods of time, over 20 days, and then only see dormant parasites persisting. And they're difficult to see because they're not high in number and they're just single parasites, but one can if one looks hard enough. And so we've developed this idea of dormancy being a problem for drug resistance or for resistance to cure by drugs. And so how do we deal with dormancy in terms of drug treatment? And there's a couple of ways you could do this. One would be to give drug continuously. So you suppress parasite development and hopefully eventually kill all the parasites as they come out of dormancy. That's not going to be a feasible thing to do with the drugs we have now because they have toxicities. One could alter the drug regimens, and we've done a little bit of that. I'll show you one piece of data on that, or develop other drugs that can more effectively kill dormant parasites themselves, or both dormant and active parasites. And this again is this is looking at the gut of an untreated animal on the left and 2 days of treatment of benznidazole on the right. And you can use light sheet microscopy to sort of cruise down the gut and see this heavily infected gut. This is a gamma knockout mouse that you don't get rid of the parasites with 2 days of benznidazole treatment, but you get rid of a vast majority of them in this very effective, with this very effective drug, but it doesn't clear the infection. And so one of the modifications that we've done is to use higher doses of the drug, but giving it only weekly. And here again, using light sheet microscopy, we're able to both identify drug failures where we see single dormant parasites in tissues, and drug success when we give high enough doses of drug over at weekly intervals over a long enough period of time. So we think there is some promise of using benznidazole and existing drugs, but really think that the real promise is in developing new drugs. And I'll close with just this slide of some of the screening that we're trying to do to look at drugs that kill dormant parasites. Don't have time to go into it in detail, but happy to answer questions about it. And the other part, immunological side of things, is what does dormancy have to do with the immune response? And that we don't understand yet, and we hope to understand it sometime in the future. And so I'll stop there. Again, the people I tried to acknowledge who did most of the work I didn't mention by name, but Fernando worked out the whole light sheet microscopy and tissue clearing business. Angel and Juan have both been critical in the drug discovery and immunology stuff. Brooke runs the lab as the research coordinator, and then Wei did all the work on the genome sequencing and comparison of the large gene families that I missed, and showed a little bit of Natasha's data on the Sting and IFNOR knockouts. So I'll acknowledge our support, which has been mostly the NIH and the Wellcome Trust, a lot of great collaborators that we couldn't do close to what we have done without them, and then more recent members of the lab that I've talked about briefly with some of their data. So I'll be happy to take questions, I think.
Okay, thank you very much, Rick, for this great talk, really informative. So we have a first question from Walderes Dutra. She says, thanks for the excellent talk. Do you know if the changes in the expression of the variants reflect different glycosylation profiles?
I missed the last part of that, sorry.
Yeah, I will repeat. So she said, if you know if the changes in the expression of the variants, the parasite variants, reflect different glycosylation profiles.
We haven't looked at that. I'm sure there is some glycosylation differences in them. It's when you have a couple of thousand genes in these families and they're being expressed, it's a little difficult to study them on an individual level. You'd need a probe for each thing. It wouldn't surprise me that there are glycosylation variants, but it's not something that we've dealt with in large part because, for the T cell, especially for CD8 T cell responses, it's not really relevant. So it's not something that we've dealt with. Sorry.
Okay, I have one question that is curiosity that relates with one of the very first slides you showed, that relates with how the cells recognize toxoplasma and trypanosoma. And I wanted to know if if toxoplasma shows more pumps or dumps than trypanosoma cruzi. I mean, it is probably more recognized by immune system, by the innate immune system.
Well, I don't know that, I'm not familiar enough with the toxo system to know if they have been officially recognized as PAMPs, but we do know that toxo, when it invades, it injects a whole ton of stuff into the host cell, right? So clearly it makes no attempt to enter quietly. It's releasing all manner of heck. And so, yeah, I'm sure some of those things are sensed. And, the other thing toxo does that I don't think, we don't have any evidence that T. cruzi does, is that it also injects suppressors of those signaling pathways. So it starts to regulate the host. But to me, that's a case where, okay, if you're going to inject, if your strategy is to come in and remake the host cell, then you're going to have to regulate the response to that. And whereas T. cruzi, I think the strategy is, I'm just going to slip in here really quietly and nobody know I'll be here. I'll leave in a little bit. Just don't, I'm not going to bother anything. That's the way I look at it. A lot of people don't like that view, but I like it.
Okay, now we have a different question from Adelina Riarte. Did you say that high doses of benznidazole are able to kill dormant parasites?
Hi, Adelina. Yes, we know that there is an attrition of dormant parasites in cases where we're giving higher doses of benznidazole. So yes, it does look like, so our understanding of dormancy is pretty poor. But I think it's tilting more and more towards the fact that dormant parasites aren't totally metabolically inactive. So they are metabolizing. And we've started to try to look at this specifically in terms of, so benznidazole, for instance, has to be activated by nitroreductase. So we'd like to know what the activity of a nitroreductase is in dormant parasites. And I think there's some ways to do that. But yes, my feeling is that if you give enough, high enough drug long enough, you can kill some dormant parasites, if not all dormant parasites, using benznidazole. But I imagine there are also parasites that are much more metabolically inactive than other ones. So dormancy is not a single state. Maybe it's a continuum, perhaps.
Okay, we have a different question from Eva Costa Rodriguez. She says, excellent talk, thank you. It is very intriguing that CD8 T cells are not close to infected cells. Have you had the opportunity to look whether the infected cells are surrounded by other immune cells like B cells, CD4 T cells, Tregs?
So the image I showed there was all T cells. So it wasn't just CD8 T cells, and we don't know. We're starting to try to figure out what T cell populations are where. So those would be CD8, CD4s, Tregs, whatever is all the T cells of what we were seeing in that one image. So we don't see any T cells that are really close there. And we need more data on what the boundaries are of an infected cell versus where the inflammatory cells or the T cells coming in are. We haven't looked at B cells. It's certainly something we would like to look more broadly at a wider variety of cells. That's just a first look using mice that were available that we knew we had a marker for T cells in those tissues that we could see. So that's one of the things that Fernando's working hard on, is starting to integrate antibody staining into that protocol so that we're not reliant just on marker mice, but we can actually look at individual populations of cells. Good question, thanks.
Okay, one more question. Nice talk. This is from Marcel Ramirez. What about data from J. Kelly group showing that crusade will be resident in intestinal cells during chronic stages? Did you check this in your experiments?
Yes, we frequently look in the gut. I think our assessment of that is that, sometimes there are parasites in the gut, sometimes there are not. It isn't, in our mind, it's not the repository for where all parasites are in a chronically infected animal. They can be in fat, they can be in gut, they can be in muscle anywhere. And when they're in the gut, they're in muscle there as well. They're not in any special other cell that allows them to be in the gut. So, we occasionally find animals where there's a lot of parasites in the gut. We find animals where there are a lot of parasites in the testis, for example, occasionally. So it's, I think there's not one place where you're going to find them. It's varied with each infection.
Okay, I have one more question. So you have shown that in your, if you modify the quality of the CTL response, for instance, or expressing the epitope par4, the one from the flagellum, you get a good clearance of the parasite. Have you checked, if possible, because I know that the tetramers against these epitopes are not really good, but do you have the chance to check a specific memory against that epitope in a reinfection? Will be this memory more effective than the transialidase TSKV20 memory?
Well, that story has gotten quite complicated. What we have found, and I could spend an hour talking about Molly Bunkowski's work, who has followed up on a lot of that. And essentially what the finding is that those TSKB20 specific T cells are highly cross-reactive to a large number of different peptides, including that peptide in the flagellum. And so that response we see there is probably not a specific response for that epitope. It's a cross-reactive response by this population of T cells. And, Molly had a very interesting project where she tried to expand. She looked for more T cell epitopes in the flagellum and found a ton of them. It was fantastic. This result was like, oh, well, we've got all these new epitopes. And then she found that every single one of them were cross-reactive with the TSKB20 specific T cells. And so now her project has morphed into understanding how that T cell response is generated and how they get so cross-reactive and what the T cell receptors are that is so highly cross-reactive. So it's another layer of immune evasion, I think, that we don't yet understand, but unfortunately it's taken us off the course that you're asking about, and we've never gotten back on that course. We've tried to identify other epitopes. Actually, Molly and Natasha identified another great epitope. It ends up being in a mucin gene family, which is also highly variant and has a weird response as well. So yeah, we wish we had other things in the TSKB20 specific or the transality specific epitope, but we don't. The long and short of it is, yeah.
And I think now Ana Rosa will do a question also.
Dr. Perleton, I have a question. Do you think that a combined therapy of besnidazole plus therapeutic immunotherapy like transialidase immunization, for example, could better control the dormant mastigotes?
Yeah, I think that's a great question still, and it's something that we do have an interest in. Is can you? I mean, one of the simple things that we're trying now is if we combine. Well, one it's difficult for us to figure out how to improve upon the TSKB20 specific T cell response, and given that it has or the transalidase specific T cells, whatever you want to call them, given that it's huge cross reactivity, and we think they're not very good effectors. But could you immune enhance the whole response against T. cruzi and combine that with drug therapy? So we are actually trying a system where we upregulate class 1 MHC, but at a much lower level and a much more subtle, and combine that with drug therapy and ask, okay, if you do those two things at the same time, can you get a better clearance? I think that's a worthy thing to look at. I'm not so optimistic, but I mean, again, we don't understand. You know, my suspicion is that dormancy is also an immune evasion mechanism. It would make sense. If you're not metabolizing, you're just sitting in a cell, the immune system's not going to see you very well. So anything you do immunologically, it's going to be difficult to change that biology. Yeah, good question. Thanks.
Thank you very much.
Yeah.
Okay, Rick, I think there isn't any more questions, so we could probably continue with the program. Thank you very much for your talk.
Thank you, Doctor.
Really informative. And now Karina will introduce Walderes Dutra.
Yeah, now it's my turn to introduce Dr. Walderes Dutra. Dr. Dutra is a full professor at the University of Minas Gerais of Belo Horizonte, where she conducts her research mainly focused on the study of different aspects of the immunobiology of Chagas disease, having published more than 100 of articles in premier scientific journals and being granted by national and international agencies. She is a leader in the field of Chagas disease. And today she will talk about her most recent interest, a special subpopulation of T cells.
Gracias, Karina. Gracias, Ana y André, por la invitación. Me encantaría mucho estar en Buenos Aires contigo, pero espero tener otras oportunidades. Y bueno, para no comprometer una nueva invitación, yo voy a cambiar al inglés. I fear that if I butcher your beautiful language too much, you're never going to invite me again to come give a presentation here. So I'm very glad to be here participating in this excellent meeting. Let me see here. So how do I share my screen with you?
Excuse me, problems with internet. Sorry, Walderes.
Oh,
Thank
sorry,
you so
Karina,
much.
you were still...
I come back, and I wanted to thank you for spreading the symposium with your colleagues in Brazil. Thank you so much. You know that you did it.
It's a pleasure, Karina. Can you hear me?
Yes.
And now you can see my screen, right?
Yes.
Okay, very good. Okay, so today what I want to talk about it's a little bit in touch with what Rick started talking about, the generation of memory and the importance of having a good memory, and especially some subjects that we've been interested in the lab currently, which deals with how important it is to keep a balanced immune response to avoid pathology in Chagas disease, and to think about how we can temper with the immune response of the patients to try and benefit and essentially avoid pathology in Chagas disease. So let's talk a little bit about it today. First, I wanted to remind you, to show some of you where we are here in Brazil, in Belo Horizonte. These are pictures from our state, and I'm showing these hoping that this will stimulate you to come visit us once we get through this pandemic. This is the Minas Gerais state. Minas Gerais certainly has the best food, hands down, in Brazil, so that's another good reason for you to come and visit us. And this is the institute where I work, the Institute of Biological Sciences at the Federal University of Minas Gerais, which is located right here near the Pampulha Lake in Belo Horizonte. And this is also for Karina to remind her times in Belo Horizonte and hopefully come and visit us at some point. So our lab has been focusing essentially in studying T-cell responses and immunoregulation in human parasitic diseases. And I am sure that a lot of this interest came from the years that I worked with Bob Kaufman at the Next Research Institute during my PhD. Bob, as many of you know, together with Tim Mossman, described the dichotomy, the functional dichotomy of CD4 positive T cells into Th1 and Th2. And it's very interesting to remember that this, which is one of the central points of immunoregulation still, these studies were done using immunoparasitology. So they were described as using a worm model and then later on using the Leishmania model. So that's a good contribution of immunoparasitology to basic immunology. So essentially, Bob and Mossman described the development of CD4 T cell precursors into Th1 and Th2 cells under the pressure of IL-12 and IL-4 respectively. And the Th1 cells were clearly associated with an inflammatory and cellular response, whereas the Th2 cells were associated with an antibody response. We know that currently the picture is a lot more complex with the description of many different CD4 T cells that have different functions based on the cytokines that they produce. And we also know that not only CD4 T cells, but also CD8 T cells and cells from the innate response can display different functional activities based on the cytokines that they produce. So we've been very interested in trying to understand these immunoregulatory networks produced by different T cells and different cells of the immune system, and how these can interfere with disease development. Particularly today, of course, I'm going to talk about Chagas disease. Like Rick said, I don't need to go into a lot of details on Chagas disease for this audience, of course, but I'd just like to remind you that after the acute phase, which is characterized by a very high parasitemia, and as was also mentioned to Rick, the parasite is really well controlled by the immune system here, and this acute phase ends and the patient enters the chronic phase where the immune system still keeps the parasite in check. And during this chronic phase of the disease, it's important to remember that most of the patients will remain in an asymptomatic clinical form that is called the indeterminate clinical form, but about 30% of the individuals will develop a very severe pathology. And the one that we've been studying mostly in our lab is the cardiac pathology of Chagas disease. Essentially, the development of the cardiac pathology or even the digestive form will be associated with the development of an inflammatory response. So we go from a form that is asymptomatic without a strong inflammatory response, despite the fact that an immunoreactivity to the parasite does exist to keep it in check. But we go to a point where this response is no longer well controlled and the pathology will get established here. Unfortunately, we don't have biomarkers that will allow for us to track this development. And essentially what we really want to do is try to understand if we can stop this transition. can we interfere with this lack of inflammation to keep it that way? Or even if the patient have started to develop the inflammation, can we work to control this inflammation? So that's the main question, really. And the way we've been approaching this is, like I mentioned to you, by studying the T-cell responses in this chronic disease, as well as the immunoregulatory networks. And a lot of work from our groups and from other groups in Brazil have associated two different profiles with the two different clinical forms of Chagas disease, in a way that we have a predominance of expression of inflammatory cytokines such as TNF-alpha and gamma interferon in patients with the cardiac clinical form, and a predominance of anti-inflammatory cytokines such as IL-10 in the individuals that don't develop clinical disease, which are associated with the indeterminate clinical form. So essentially, a lot of studies showing that the expression of different cytokines by T cells, CD4 T cells, CD8 T cells, and innate cells are shifted towards an inflammatory profile in cardiac and symptomatic patients, and are more balanced in the indeterminate patients. One interesting aspect when we were interested in trying to define the sources of these cytokines, and the reason why it's important to define the sources of the cytokines is because in order for you to interfere with this response, you need to know the cells that are producing the cytokines to be able to control somehow their activation and then alter their expression of different cytokines. So when we were starting to look at that, one of the interesting things that we found was that looking at the contribution of CD4 T cells and CD8 T cells to the expression of the inflammatory cytokine gamma interferon, we found that over 55% of gamma interferon in cardiac patients did not come from CD4 or CD8 T cells. And interestingly, looking at the expression of IL-10, which is the anti-inflammatory cytokine, in cells from indeterminate patients, over 70% of IL-10 did not come from CD4 or CD8 T cells in the indeterminate patients. So that led us to think that this subgroup of T cells that are called the double negative T cells could be important in this response. So who are these double negative T cells? The double negative T cells, as their name says, are T cells that do not express the co-receptors CD4 or CD8. They comprise about 2 to 4% of the T cells in the peripheral blood of humans. They can express the alpha beta T cell receptor or the gamma delta T cell receptor, just like the regular T cells. But the antigen recognition of these double negative T cells is very much related to the expression of these molecules by antigen presenting cells that belong to the family of CD1 molecules. So I put here 4 different CD1 molecules that can present antigens to double negative T cells, CD1A, B, C, and D. These cells, upon activation, are able to respond fairly quickly and fairly intensely, and they tolerate chronic stimulation because they don't have the co-receptor, so that's very helpful. Importantly, in the case of Chagas disease, these T cells recognize mainly glycoconjugates. And as we know, it's been described many years ago, the surface of T. cruzi is decorated with a huge number of different glycoconjugates that would be important in the activation of these T cells. So the first studies that we did in the lab regarding the contribution of double negative T cells to cytokine expressions in Chagas disease by Fernanda and Livia had shown that these cells can indeed produce both inflammatory and anti-inflammatory cytokines in patients with both different clinical forms of Chagas disease. So these double negative T cells express gamma interferon, TNF-alpha, IL-10, and IL-17 upon activation in vitro. But interestingly, when we looked at the ratio of anti-inflammatory and inflammatory cytokines represented here by IL-10 and gamma interferon, what we observed was that this ratio was shifted to an inflammatory ratio in the cardiac Chagas patients, but the indeterminate asymptomatic patients had a more balanced profile between inflammatory and anti-inflammatory cytokines produced by these cells. So these cells were robust producers of cytokines, but they did have a different profile if you compared the two different clinical forms of Chagas disease. So the next question came at the time at the lab, in the lab, which was, if we block the activation of these cells, could we switch this profile of inflammation seen in the cardiac patients? And to do that, Livia first showed that the activation of double negative T cells in cardiac Chagas patients was directly related to the expression of this particular CD1 molecule, which was the CD1D, in which she's found an association of expression of CD1D with production of gamma interferon and with the expression of activation markers in the double negative T cells. So our approach to respond to this first question was to take cells from cardiac Chagas patients, stimulate in vitro with live triple mastigotes of the Y strain, and block this interaction of CD1D with the double negative T cells using anti-CD1D monoclonal antibodies. And then the readout was the expression of activation molecules and the expression of the two cytokines, one inflammatory and one anti-inflammatory, and we did all that using flow cytometry. And what we found was that after blocking using the anti-CD1D antibody, she found the decrease in the expression of the activation molecule CD69 in double negative T cells, particularly of the gamma delta subset. And importantly, this, as expected, had no effect on CD4 and CD8 activation, which might be very important to keep their functions to control the parasite, as Rick mentioned. So after that, we looked at the expression of the two cytokines to see if the blocking would have any effect. And what Livia found was that there was no alteration in IL-10 expression by the double negative T cells after the blocking of their activation, but there was a decrease in the expression of gamma interferon in the cells from the cardiac patients, therefore shifting this balance to a more well-controlled, well-balanced immune response. So we are now asking ourselves if this would have a potential for therapeutic intervention. And at this point, what we really wanted to do was to find out within the double negative T cells, who were the subpopulations of memory cells that were responding to this activation in vitro and being affected by the blocking of this activation. And the reason for that is because when we study the activation of the cells in vitro, essentially what we are seeing is a recall response that will reflect the response of activated memory cells that are present in the patient during this chronic infection, right? So Salusto and Lanzavecchi a long time ago have divided the memory T cells that arise from the activation of naive T cells in the case of trypanosoma infection after the contact with the parasite into central memory cells and effector memory cells. The difference between the two of them are many. They have different phenotypic profiles, expression of different proteins, different functions. For example, the effector memory cells are capable of proliferating quickly, and they home directly to the site of infection and can respond also very quickly. Whereas the central memory cells generated after this first contact with the antigen are very important for maintaining the response, but they need a secondary stimulation to be able to respond. The issue of memory in Chagas has been studied by different groups, especially the group of Susana Lausela in Argentina, with the papers by Albareda, discussing the memory characteristics of CD4 and CD8 T cells and how the cells can get exhausted after a long chronic stimulation. And also in Brazil, by Rodrigo Corrêa Oliveira's group, Jaqueline Fiuza, discussing the memory characteristics of CD4 and CD8 T cells, finding a little bit different data, but showing that these cells are different in indeterminate cardiac patients. So our question really was, comparing these different populations of memory cells, what are the ones that are responding when we look at the whole double negative T cell population? And again, the idea of identifying them is just so we can gain more knowledge into these populations to see how we can manipulate them for benefiting the immune response. So to analyze that, we also took advantage of the paper by Federica Sallusto and Lanzavecchia, and the strategy was to take cells from indeterminate and cardiac Chagas patients, expose in vitro to live Trypanosoma cruzi from the Y strain, and then evaluate activation markers and cytokine expression into the different memory populations. So here, first we select the cells that do not express CD4 or CD8, but do express the T cell receptor alpha, beta, or gamma delta. And then we use the expression of two surface molecules, CD45RA and CCR7, to define the different subsets of memory population that I mentioned before. And then within each one of these different populations, we can evaluate activation markers and cytokine expression to get to know better about the cells. So this plot here shows the expression of CD45RA and CCR7 and how we can let us subdivide the different populations into central memory and effector memory, but also naive cells and effector cells. And by doing the differential analysis of expression of these molecules, we can separate them into different populations using this strategy here that takes under consideration the expression of all the molecules in the pool, which is called the t-SNE analysis. And after we evaluate the t-SNE analysis based upon the expression of these markers, we can come with a plot like this that allows us to identify, for example, a population of central memory cells right here that has a low expression of CD45RA and high expression of CCR7 right here. The more yellow, the lower the expression, the more red, the greater the expression. So they have low here and high here, and they are located in this picture here. So this defines to us where these different populations can be found when we use a t-SNE analysis. And when we did that in cells of cardiac and indeterminate Chagas patients, what we found was that indeterminate patients had a predominance of this population right up here that we know is referring to the central memory population, whereas the cardiac patients had a predominance of this population down here, which is the effector memory population. And when we went on to quantify that in all of the patients that we analyzed, this is clearly shown here that the indeterminates have a predominance of the central memory, which is showing on the side of the graph, as compared to the cardiac patients, either before or after the stimulation with triple mastigotes in vitro. And that happened in the double negative T cells of the alpha beta and the gamma delta TCR. And the opposite was found when we quantified the effector memory cells, which is higher in the cardiac patients as compared to the indeterminate patients in both subpopulations of double negative T cells. When we now look at a ratio of central memory over effector memory in indeterminate and cardiac patients, this is basically done using the same data that is here, but just to show you that really there is a predominance of central memory in indeterminate as compared to cardiac patients in both subsets of double negative T cells. This here shows a clinical correlation between the frequency of the two different memory populations in double negative T cells, which are the central memory and the effector memory populations, in relation to a function that reflects a cardiac function in this patient. So this is the left ventricular ejection fraction, which is essentially the ability of your heart to pump blood. So the higher the number, the better the function. And as you can see here, there was a positive correlation between the frequency of the central memory subpopulations in alpha, beta, and gamma delta double negative T cells with a better cardiac function. So the more central memory, the higher the ejection fraction. And the opposite was observed for the effector memory in a way that the higher the frequency of the effector memory, the lower the ejection fraction, suggesting a correlation of this frequency of these cells also with a clinical parameter. So we became interested in now this dissecting a little bit on the functional characteristics of the central and effector memory T cells. And what we did first was to measure the cytokine that correlates with the inflammatory response and the cytokine that correlates with the anti-inflammatory response. and in the double negative T cells of the effector memory phenotype. And what we saw was that whereas a stimulation in vitro with triple mastigotes increased the expression of gamma in cells from indeterminate of the alpha beta phenotype for the double negatives, it happened in both populations for the cardiac patients. And interestingly, there was a significant decrease of the anti-inflammatory cytokine IL-10 upon stimulation in vitro, only in cells from the cardiac patients. But the effector memory T cells, the double negatives, can produce both gamma and IL-10, but a decrease in IL-10 in the cardiac patients. With regards to the expression of these cytokines in the central memory population, what we found was that there was an increase in the expression of gamma interferon in cells from indeterminate patients after stimulation in vitro. But there was also an increase in the expression of IL-10 in the cells from indeterminate patients. It seems that the central memory cells are much more responsive when they come from indeterminate than when they come from cardiac patients. And we are trying to understand a little bit more why that is. And we'll touch a little bit on that, but there's still a lot of work to be done here. So this is interesting because we found the expression of both cytokines in this subset of memory cells. And going back in the literature, we found that Del Preti in 93 and Dragana Jankovic at the NIH in 2007 described a double expressor population of T cells that produced gamma interferon and IL-10 as being a self-regulated population of T cells that are associated with better outcome of inflammatory diseases. So we evaluated the double expression of these two cytokines to evaluate this population described by Dragana, and we found that, in fact, the central memory population in indeterminate patients had a high frequency of double producers IL-10 and gamma interferon, which was not seen in the central memory cells from the cardiac Chagas patients, suggesting that the cells are indeed functionally different in the different clinical forms of Chagas disease. Several papers had shown that T cells sometimes can produce cytokines, but they don't always express activation markers. Karina recently that sometimes showed that sometimes they produce cytokines, but they don't proliferate. So we went on to look at the expression of activation markers in the central and effector memory cells from cardiac and indeterminate patients. And this is the plot to remind you where they are. And what I show here to you is that the expression of CD69 in this Tisney plot is clearly overlapping with the location of the central memory in the indeterminate patients, whereas it's overlapping with the effector memory population in the cardiac Chagas patients, showing that they can indeed express these activation markers. And one interesting point here that you can see is that this population really is very decreased in the patients with the cardiac clinical form. So the central memory really seems to be decreased in these patients here. And when we went to quantify this in all of the patients, this is clearly shown here, where we have a decrease on the expression of CD69 in central memory cells and in central memory cells from cardiac patients after the recall activation in vitro, showing that these populations really seem to be either not getting activated or from the plot before dying. And one interesting aspect is that if you look now at the co-expression of this activation marker in IL-10, you can clearly see this decrease here in the cardiac, in the cells from the cardiac patients, but they are present in the indeterminate patients. So the CD69 cells that are decreasing in the cardiac patients are the ones that are expressing IL-10. So we believe that this will all contribute to the inflammatory profile that is predominant in these individuals to decrease, let's say, the ability that these patients have to control the inflammatory response. We have to remember that it is very important to maintain an inflammatory response, an activated response, to be able to control the parasite, but it seems that this ability of modulating this inflammation may be compromised in these patients here. When we look now at a trajectory map, looking at the expression of the different markers in cells from cardiac and indeterminate individuals, and the way we read these trajectory plots is that you have naive cells here at the top, and these naive cells, the association with them into the development of the different memory populations is shown here. You can clearly see that this is shifted to the effector memory population in the cardiac patients, and this is clearly shifted to the central memory population in the individuals of the indeterminate clinical form, showing very clearly that there is a different trajectory of the expression of these markers and these different memory populations in patients with the different clinical forms. So the questions that we're trying to address now is whether if there is a if the differentiation of the central memory populations are compromised in the patients with the cardiac clinical form, or if these cells are somehow dying. One of the approaches that we are taking now has a lot to do with the huge amount of work that Susana Lausella, Tarleton, and others in Argentina have done, which is evaluating the exhaustion in the double negative T cells. And of course, the idea would be, could we rescue these cells from activation if we can identify which population, which seems to be the central memory, to restore a more balanced response in the cardiac Chagas patients. So although we are still a lot far from a direct application of this, it seems that because the cells are susceptible, as I showed you in the beginning of the talk, to a manipulation that can change their function, we are excited about the possibility of checking this in the central memory population of double negative T cells. We recently, together with Igor Almeida, have identified the components from the parasite that stimulate the double negative T cells, and they are mostly glycoconjugate fractions from Trypanosoma cruzi. So it's very excited to be able to find a molecule that will allow for us to modulate the function of this particular and important cell subpopulation in Chagas disease. So I end here by thanking the collaborators of our work, Maria do Carmo Pereira Nunes, who is the cardiologist responsible for following up all the patients that we work with, Ken Golub and Liz Antonelli for the initial memory studies in Leishmania and in Chagas disease. The group in the lab that started the studies, Fernanda Villani, Luisa Magalhães, more recently, Lívia Passos and Carolina Có, that have been devoting their time to studying the double negative T cells in Chagas, and also Juliana Stanislau and Rodolfo Giunchete, who I share the lab with and deal with the everyday issues that are not easy in Brazil, as certainly most of you know. And of course, the funding agencies, the National Institutes of Science and Technology in Tropical Diseases in Brazil, CAPES, CNFQ, and FAPEMIG, which I truly hope can step back into funding our research, because in the past 2 years, as you know, we've been having serious issues with funding for research in Brazil. So something has to happen for us to be able to get that back. But thank you very much to the NIH, who's been funding our research. So thank you to that. And I'd like to finish wishing everyone a happy International Women's Day. I put pictures here of inspiring scientists that have been making our lives exciting and a lot better. So thank you so very much for this invite. And I'm glad to answer any questions that you might have.
Thank you, Wald, for your talk. Excellent. And thank you as well for being in these slides. It's too much for me. Thank you so much. Here I have a question from one person of the audience that I cannot recognize. Said, excellent talk, thanks. Thanks, Dr. Walderes. My question is, during the intracellular parasitism by Trypanosoma cruzi in chronic phase of the disease, how do you think is the maintenance of the activation of the double negative T cells via CD1D molecules?
Yes, that's a very good question. Yeah. So as I mentioned, the parasite has a number of molecules in its surface that can lead to the activation of these double negative T cells, yeah? So we know that the parasite is present in the host, we know it's there, we know that it is triggering a response, and we believe that either this could be presented to the double negative T cells, you know, by regular APCs, but we also suspect that the release of extracellular vesicles could have an important role in the activation of these cells. This is an issue that my collaborator Igor Almeida and Ana Cláudia Torrecillas are trying to address. But we really think that these glycoconjugates that are released somehow, we don't know what the mechanism really is yet, are the ones that are going to be activating these different cell populations.
But question from mine. But this glycoconjugate has a special core. I know that there are preliminary results and maybe you don't want to talk about that, but there is a special core of glycan.
So all I can tell you, and perhaps the glycobiologists in the audience could help us respond to that question, but what I can tell you regarding the fractions that better stimulate the double negative T cells now are two very important things. So what Igor did, so the first studies that we did, we took the parasite, we fractionated it into proteins, neutral lipids, and glycoconjugates. And the stimulation of the double negative T cells was in this third group of molecules. So Igor took that and fractionated that further into 6 different fractions based on the elution of the columns using different concentrations of solvent. Yeah. So what I can tell you is that if you take these 6 fractions that he performed, that he separated, and look at the response of the double negative T cells in the indeterminate and the cardiac patients, it seems that they respond differently to them. So for example, the fraction that we call 20% is a fraction that better stimulates cells from indeterminate patients. The fraction that he calls 40 and 60%, which is the last ones to be released, are the ones that stimulate cells from cardiac patients, which is nice. We like to have the results that can allow for us to manipulate differently. But now what we need to do is to take those fractions and go back and see what exact molecules are present there. But that's for Igor to do. That's for the glycobiology part.
Very interesting. Another question here. Thank you very much for this great talk. I would be very grateful if you can tell us about the immune response in the amastigote phase. It's a question from Anaí Pérez.
That's a very important question, Anaí. In fact, a lot of our work done with the patients in Brazil, as you know, has many differences as to the work that has been done in Argentina by many groups in Argentina using a mastigote antigen. So we've been working with triple mastigote antigens. We haven't yet looked at the response of amastigotes in our lab, and that's an important thing to do. It's a thing that we have in our plans on doing. It's just that we started doing all of this work, and especially with the double negative T cells using triple mastigote antigens. We believe that there may be critical differences in the response. And in the past, I have actually discussed this with Suzanne. I don't know if she'll remember when we met a few years ago in the Top Med in the United States. And I think this is something that we should do, you know, comparing the response of the Argentinian patients with the Brazilian patients. We know that the many differences exist, ranging from the genetic background of the different populations, the Brazilians and the Argentinians, but also regarding the different strains of the parasites that infect our patients. But also I think that the type of antigen that is used is an important point that we need to address.
Another question, very nice talk. Very nice talk, thanks. My question is if you have evaluated this subset of cells in patients after prednisone treatment, is there any change in the phenotype before and after the treatment, if you do?
That's a very good question, Cecilia. So we are in the process of doing that. So we are now just recently entered into a project with Esther Sabino in São Paulo. This project is being carried out here in Belo to treat indeterminate patients with the different benzonidazole regimens. And our part in the study is exactly to evaluate what happens to the double negative T cells after these patients are treated. So this was supposed to have started last year, but as you know, everything that was supposed to start last year was pushed forward, to this year for obvious reasons. So we just started the collection of the time zero from the indeterminate patients that will be treated. This treatment should be starting soon, and we will have 3 time points in which we'll get material from the indeterminate patients to evaluate the expression, what happens to the double negative T cells. It will be very interesting to see if they change their profile, if there is any significant changes in them, or even if they can be used as potential markers for trying to help to identify what happens with the treatment. As you know, this is a huge issue with evaluating efficacy of treatment in Chagas. We don't have a good correlate of cure yet, so we are hoping that we can also try to contribute in that area. Thank you, Pilar.
Now a question from Pilar Aoki. Do you think that the self-regulated immune profile in the double negative T cells is a cause or a consequence for a better cardiac function? Is the frequency of a double negative T cells profile related to the parasite load in patients?
So these are both excellent questions. And the response to the first one is that it's very, very difficult to know when you study a human disease that is so chronic, that of such a long duration, and with such a spectrum of diseases ranging from indeterminate to mild cardiac to severe cardiac, to find out when you have an immunological change, if that's going to be associated with a cause or a consequence. What we can say is that we associate, this population that is self-regulated, the double producers of gamma and IL-10, is more frequently found in patients with the indeterminate clinical form. Now, we don't know if when that balance is lost, or when that population, for example, just to take this as an example, if when they change their profile to no longer become self-regulated is when the cardiac disease happens, or if a cardiac alteration or an issue related to the parasite itself is what causes that change in the cell population. So it is a great question, but it's very hard to identify whether it's a cause or a consequence. And regarding parasite load, we were having a very nice discussion this last Friday. I was actually examining a thesis from John Kelly's lab, who was mentioned here earlier during Rick Tarleton's talk, discussing this issue of defining parasite load in the chronic patients. One thing that we can say in our patients here, so the short answer to your question is yes, we don't have a good measure of parasite load in the chronic patients. One thing that I can tell you is that if I take hemoculture studies from indeterminate and cardiac patients, we know that the time that is required for the hemoculture of a indeterminate patient to become positive is a lot greater than the time that takes for a cardiac hemoculture to become positive. Of course, that could be related to the parasite load, but of course it could also be related to a characteristic of the parasite itself that we don't know yet. So very, very complex issues, but very important points as well.
My last question, do you measure interleukin 17 in the double negative T cells?
Yes, Karina, we did. So the double negative T cells are also excellent producers of IL-17. I know you're interested in IL-17 in the context of T cell response and also B cell response, yes. But the double negative T cells do produce quite a bit of IL-17. In fact, they produce, you know, everything that we measure, they're excellent producers of. What changes really is the balance. So IL-17 doesn't seem to change much when we block the activation of these double negative T cells using anti-CD1D, which is what we saw with IL-10. So in our studies, it seems that IL-17 looks more like IL-10 in terms of its production, and you know, if it's higher or lower in indeterminate and cardiac patients, than it does as to comparison with the inflammatory cytokines, for example. But when we block the activation, we didn't see many changes in IL-10 expression.
Thank you so much, Walderes, for being with us.
Pleasure.
And now we can go ahead with the next talk. Thank you so much again.
Thank you. It's a pleasure.
Okay. Now, with a great pleasure, I introduce our third speaker, Dr. Juliana Demess. Juliana is graduated in biological science and have a master in cellular and molecular biology and a PhD in parasitic biological from the Osvaldo Cruz Foundation, Fiocruz, in Rio de Janeiro, Brazil. Later, she did a postdoc in the Institute of Biophysics, Carlos Chagas Filho. Currently, she is a field researcher of the Laboratory of Thymus Research at the Osvaldo Cruz Foundation, and is also a permanent professor and member of the post-graduation committees of cellular and molecular biology and bioscience and health, both from Fiocruz. He has a lot of experience in the field of Chagas disease immunology, with a special emphasis on oral infection, apoptosis, telangiectasia, regional immune response, inflammation, and hemostatic disorders. She has more of 30 referred publications in the area of Chagas disease. At present, he is contributing to health policies in the acute and chronic human disease in the state of Pará, in the north of Brazilian Amazon, and developing translational studies in oral Chagas disease. Today, he will talk about studies on target organs and the host immune response to oral T. cruzi infection. So, Juliana, we are really happy to have you here in this symposium, and thank you for sharing part of your results. If you want to share your presentation.
Thank you, Anna. I will share now my video. Just please tell me if you can see it. Can you see it?
Yes.
Okay. Now I want to thank the organizers for the invitation. I was really happy to be in this very interesting symposium with so many important immunologists of Chagas disease, like Tarleton and Walderes and my colleagues. And I will try to give you a talk a little bit different from those who were talking before me. I will talk about immune response, of course, because this is the topic of the symposium. But first, I have to introduce you this oral infection. What is this? Because if you see in books and in the CDC, everybody talks about the vectorial route of infection, but the oral route of infections is somehow forgiven. And now we have to sensibilize you about this route of infection. So in Brazil nowadays, the most important route of infections are the vectorial route, as Tarleton and my colleagues already showed, but we also have the oral route of infection. Nowadays, in numbers of notification for our health ministry, these are the most important one. The oral route is involved in consumption of meal with parasites, contaminated with parasites. So people usually can eat wild animals, which are reservoir of the parasite. And if they eat this meal, they can be infected. And it does happen a lot in the north, in the Amazonic region. For example, people usually go to the forest to get their meat to have their lunch or dinner, and they are susceptible to oral infection. And also the vector can be crushed together with vegetables and fruits, and people can get infected by drinking these contaminated juices. So in Brazil, we have description of oral outbreaks in several states. This is not a recent map, but you can see here that almost every state had already described oral outbreaks of Chagas disease. And this is an important thing because in oral infection of the parasites, one important thing is that it usually comes as an outbreak because people usually share their food. And if this food is contaminated in the dinner, in a family dinner, or in a party, everybody is infected with that food. So numbers are really high. In 2019, we had an outbreak next to Recife with more than 70 people at the same time being infected orally by Trypanosoma cruzi. So I want to show you that oral infection can be related to consumption of vegetables, for example, crushed with the vector. If the vector is infected, it will contaminate these vegetables. And it's the most common route of infection in Brazil. Also, wild animals are able to eat these vectors. And if the man goes to the forest and take these wild animals to eat, it can be infected as well. And we know that mucura in Argentina and gambá in Brazil, and the delphis are reservoir of the parasites, and in their feces and the excreta, they can release the parasites, and they are in the forest, so they can contaminate fruits and vegetables as well. And it's still an open question if mother can pass by milk for the kids. The most common fruits in Brazil are the açaí, a very famous fruit here in our country. It's cultural. Our people love to eat açaí, and as fresh as the açaí is, as much the people want to eat it. And açaí for itself, it's able to release CO2, and the vector goes after the açaí and can be infecting these fruits, bacaba, babaçu, and sugarcane. So according to our governments, we have outbreaks of oral infection all over the country. This is from 2000 to 2013, okay? And I want to stress this state, the Pará state, which is in the Amazonia region, Brazilian Amazon region. In this state, we can see that we have more than 800 cases of oral patients in this period, 2000 to 2013. And as the outbreaks occurs with several people at the same time, these numbers get growing and growing and growing. since the government started to look at this route of infection, because until 2005, 2006, nobody was worried about this oral infection. But after that, people started to look at this route of infection, and they started to find a lot of cases, and the numbers are still growing up, okay? So we decided to go to Pará State in order to work with them, to help them, and to try to understand this route of infection, to see if it's the same route, if the immune response in these people are similar to any kind of route, like vectorial routes or anyhow, vertical routes. We decided to study these patients and to help people in the States to go after this new route of infection. So in this study, we were working just with secondary data, just to show you that in Brazil, it's compulsory, the notification of acute cases. So in 2000 to 2016, in the Pará state, in a single state in the Amazonia region, we can see several cities that reported cases of acute Chagas disease. You can see that some cities concentrate these cases and some cities does not have Chagas disease notification, which is quite strange since the habits, the cultural are similar in all the states. Here is notification cases from different routes of infection. So these are, at the moment that the patient is notified in the system, they tell what was the possible route of infection. So the oral route is in blue, the vectorial route is in red, other routes, which means they don't know which route was, and this in yellow is the number of deaths. We can see that after 2007, only in Pará state, about 200 people started to be notified in 2009, and now in 2016, we have almost 300 people being infected orally in this state, meaning that this is really a health problem in the Amazonic region and in Brazil. I just want to tell you that this is not a problem from Brazil, because Venezuela, Argentina, Ecuador, and Bolivia already described outbreaks of Chagas disease by oral route. So it's our problem, it's the problem of countries with the vectors, with infected vectors, okay? So we started to work there by talking with the people, giving classing, helping the technicians, microscopists, and doctors to identify those patients in order to identify and understand how is really happening this route of infection in the state. Because if once it was silenced, now we have to tell the people that they have to look about, they have to worry about, and we need to understand what's happening there. So we went to the field, we talked to the people, we look at patients. They have patients, I showed you more than 200 per year. And we, with our contact with these patients, we started to make questions to answer in the lab. So we start with different thinking. I love hard immunology. I love the questions, immunology questions, but now we changed our thinking. We started to look at the patients, see the problems in the patients, and then go to the lab and trying to answer them. So these, I think maybe you, I don't know, I have this in front of me. Can you see this that I'm showing? I don't know if, okay, because there's something happening here, okay. So the first thing we wanted to analyze the main symptoms of these patients. So we had this very good collaborator, Dr. Dilma. She's a cardiologist from Pará. She's a reference in Pará state for Chagas disease. And Valeria, she was a student, a PhD student that lived in Pará. She helped us to understand what was going on with these patients in oral outbreaks. So as you can see in this table, the most common symptoms are fever, asthenia, facial edema, headache, myalgia, abdominal pain, and hepatomegaly, splenomegaly, which are very common in some of our subcutaneous infected patients, were not so common in these patients, suggesting that there are some differences in some symptoms of these patients. I don't know why it's not going. Sorry, people, I think I have a problem. Are you there? Can you hear me?
Yes.
Yes.
Yes, yes.
But I don't know why it's not moving here. I think I have a problem in my computer, sorry.
No problem, take your time.
What's going on? It's not moving.
Maybe you close and open again.
Yes, I'm trying. Let me see.
Yeah, it happened to me the same.
Yeah, in the middle of my talk.
I can try to stop your presentation and then maybe you can try to
Can
load
you
it.
please do that for me?
Let's see if I can.
Because I can't move anything here.
You cannot, Andres?
No.
I'm trying.
Yes.
Okay.
Okay.
I think I
Thank
did
you.
it. Fine.
Disable participant screen sharing. Okay, can you enable again?
Yes, I guess you can try now.
Okay. Oh yeah, thank you. So the first point that we wanted to talk about is how is this oral infection? I mean, is this one? I remember someday I was talking to Ana Rosa when she was here in Rio, and she said, Juju, maybe it's the similar. It doesn't matter the way the parasite goes inside the host. The infection might be the same. So this was our first question, and Ana itself helped us to answer this question. So we look at the literature. There was only few papers in the literature working with oral Chagas disease. And there is this very good scientist from USP, Nobuko Yoshida. She always performed intragastric infection. So she puts the parasite in the stomach of the mice. And she at times suggested that the main target cells are the epithelial stomach cells. Since the parasites were able to interact with mucins in the epithelial cells, and infect these cells and then goes to the circulation. There was also this Hoft study, very interesting, which he was infecting the animal and he was cutting the animal everywhere. And he looked at inflammatory filtration in several tissues in infected animals. So this was the main points that we were working on the literature at the time we started to work. So we decided to answer the question, what's the first target organ after an oral infection? Is it really the stomach? Because at the moment that people drink or eat something, the parasites go to the mouth, and after that goes to the stomach. For that, we took advantage of these bioluminescent studies with luciferase triple mastigotes, and we put it in mice to drink these parasites. As we can see here, after 15 minutes of drinking the parasites, most of the parasites were located in the head, which was quite a surprise for us. It was not in the stomach. And after 60 minutes, these parasites were still in the head. And after 7 days and 21 days of infections, they were already in the body, the whole body. They got the circulation and they were found in other tissues. So this data suggests us that the main target tissue may not be the stomach, but in the head, okay? And when we look at PCR analysis, we observed that at the nasal cavity, here's the open mouth of a mice. The nasal cavity was the main target tissue of infection. So after 60 minutes, the number of parasites in the nasal cavity was high. After 7 days, this parasite was really proliferating a lot. And after 21 days, they were still there. The esophagus and the stomach show us that they were not the main target tissue in the beginning of the infection, but yes, they were a target tissue after the parasite was systemic, as well as the intestine, the mandibular lymph nodes, and as you can see in the spleen, liver, pituitary gland. This is an important thing. We noticed that in bioluminescence analysis, the parasite was migrating from the nasal cavity to the brain. In patients, indeed, acute patients have problems of face edema and memories, depressions, and signs of nervous system alterations, central nervous system alterations. Okay, so working with this method, we answer that oral infection, the main target tissue is the oral cavity, it's not the stomach, as it was before thinking. And at the moment that the mice eat something with the parasites, there is a high infection rate and proliferation in the nasal cavity. And after this proliferation, this parasite goes to the blood and starts to infect the heart, salival gland, and other tissues, peripheral tissues, but also migrates by the olfactory bulb to the brain and infects the brain, which means there is a possibility that this infection may target the central nervous system very hard. So in fact, when we had these results, we were thinking, but most of the studies, if you go to the PubMed and put oral infection of any parasites, you will see that usually authors consider intragastrical infection as an oral infection, because it does make sense. If you put in the stomach, it goes by the digestive tract. So we answered, if we put the parasites in the mouth or in the stomach, is the immune response the same? Is the disease the same? Does the disease develop at the same way? So in this study, Juliana Barreto de Albuquerque, she was making her PhD, and we performed this experiment. We infected mice in the mouth or in the gastric, intragastrically, okay? And we looked for the development of the disease and the immune response. As people already talked here, we know that several cytokines are important to control or not to regulate the immune response. And these cytokines are also important to control the tolerance and the parasite escapes and parasite killing. So we know that Th1 cytokines like interferon, TNF, and IL-12 are important to activate macrophages and helps to kill the parasites. So decreased parasitemia, decreased parasite load, but increased damage in the heart. It was already well described here by our friend Walderes Dutra. IL-4, IL-10, TGF-beta, and IL-17, in contrast, they are not good to help to kill the parasite. In contrast, they help the parasite to proliferate inside macrophage, for example. They improve the parasitemia, improve parasite load in tissue, but decrease damage in the heart in target tissues. So we want to ask it, how is the cytokine profile in these two types of infection, if you put the parasite intragastrically or in oral infected mice? For our surprise, we can see here in black is the oral infected mice. So mice that drink the parasite, and these are the mice infected in the stomach, okay? We can see that parasitemia is higher in the mouth when the parasites are put in the mouth. And mortality is also increased when we put parasites in the mouth, in contrast with the stomach. And people may ask, but is this related to the stomach acidity? Because if you put in the stomach, the pH may kill the parasites. So Nobuki Yoshida already showed that the parasites are resistant to the pH of the stomach. And we performed this experiment where we neutralize the acid pH on the stomach. And we can see that mice infected orally were having the higher parasitemia when compared to mouse infected intragastrically. By looking at target tissues, it was very interesting. We noticed that when we infected mice intragastrically, the heart seems to be worse than mice infected orally. It was a quite surprise because we were thinking, if these mice were dying more, the heart would be worse. But in fact, orally infected mice who were dying had better hearts, less infiltration in the heart than mice infected intragastrically. But in contrast, the liver was different. By looking at the liver, we noticed that the liver of orally infected mice were with a lot of infiltrating cells that were not observed in intragastrically infected mice, suggesting that the route of infection may interfere in the evolution of the disease and even compromise some tissues instead of others. By looking at liver enzymes out-nest, we can note that indeed orally infected mice have higher enzymes, suggesting higher lesion in the liver than intragastrically infected mice. And what about the serum? How is cytokine in the serum of these two groups of mice? As we can see here, in black, these black bars are orally infected mice, and this blue bar are intragastrically infected mice. In oral infected mice, it seems that Th1 cytokines are higher along infection in the serum. And in intragastrically infected mice, IL-17, IL-10, and TGF-beta and IL-4 are more important, more prominent, suggesting a different pattern of cytokine response. In heart, which is a target organ of the infection, qPCR of this tissue showed that indeed orally infected mice have higher Th1 cytokine production in the cells, and in contrast, in intragastrically infected mice, TGF-beta was compensating this Th1 response, and IL-10 was different. And so we asked, which is the most most important cell producing these high levels of TNF. If you can see, after 17 days of infection, the levels of TNF were very high. So we noticed that in the heart and in the liver, in both tissues, the main producing cell is the macrophage. Macrophages are the main producer cells of TNF in the heart and in the liver in both cases. I can show you everything because there's a lot of data. I can answer questions after that. But this work suggested that when we put the parasites in the mouth, the infection is worse. There is a huge Th1 immune response, and this is compromising the life of these animals. So by going back to the patient, when we were working on the field, talking with doctors and talking with the patients, we noticed that an important thing that happens with some patients are hemorrhagic manifestations. So these patients start to bleed. And at the beginning, they were not paying a lot of attention with that because they start to bleed in the mouth, in the nose, and some other places. There are severe cases of hemorrhagic in the gastric area, intragastric hemorrhage. So we asked, is this hemorrhagic, this bleeding related to immune response? Now in COVID infection, everybody's talking about storm of cytokines and thromboembolism and alterations in the coagulation systems. Before the pandemic, before COVID-19, we asked these questions for Chagas disease. So this is just to show you, these are our manual for clinicians to localize, to suspect of acute case of Chagas disease. And both things they are always telling the people, the doctors need to know, you have to look at out and ast because probably there are liver compromising in these patients and hemorrhagic bleeding in these patients. So this is a case of an outbreak in Ceará. This was described in 2009, and we can see that this patient showed hemorrhagic signs and thrombocytopenia. So we asked, is the immune response, this pro-inflammatory immune response related to these alterations in hemostatic behavior? Why are these patients bleeding? What's going on? So Dina was in her PhD in our lab, and she tried to answer this question. So she infected mice with less virulent strain of the parasite, so it won't kill the parasite. And we infect these mice in the oral cavity, and they got infected, as you can see here in their parasitemia, okay? Then she looked at Th1 pro-inflammatory cytokines. She looked at TNF. She saw an improvement of TNF during infection, IL-6, and gamma interferon. She also noticed that these mice, orally infected mice, showed thrombocytopenia in the middle of the infection. But after some time, since the mice was not dying, they start to recover the number of platelets. In an assay of bleeding, this is a bleeding assay for people who works with coagulation. So what do? We take the mice, we cut the mice and put the mice to bleed, and we count the time that these mice were bleeding, and then you look the number of erythrocytes that were in this liquid. So if the mice stopped to bleed fast, there will be less erythrocytes, and if the mice start to bleed for a long time, the bleeding will lead to a lot of erythrocytes, and it will change the color of the PBS. This is a saline PBS, okay? So as you can see, as much as the time passes, our infected mice start to bleed more. And we perform a lot of experiments. I won't show you everything because of the time, but we performed time of several factors. We looked at factor 5, 7, 8, factors of the coagulation cascade, time that this bleeding was taking to coagulate it. So we had a lot of exams showing that as long as the mice were infected, there improved of bleeding time in these mice. And I think you might be already heard about the dimer because of COVID as well, which is a level of, it's a sign of intravascular coagulation. So as there are fibrin released and coagulation, there is a dedeme release on the blood. So in normal situations, we don't have dedeme in the blood. The levels are very low. In mice, you don't have at all. And if you have alterations in coagulation system, dedeme starts to appear in the blood. And we can see that infected 3 in 14 days, 3 from 6 mice and 2 for 6 mice in 21 DPI was showing signs of intravascular coagulation. So we asked, okay, so these mice are having problems of coagulation, but is there any relation to the immune system? Is this because of the inflammation, because of pro-inflammatory inflammation of these infected mice? So we started to block some cytokines in order to understand if we block cytokine, we can block this increased time of coagulation. And we tried to TNF, we didn't see any change. So probably this is not because of TNF secretion, but for our surprise, IL-6, at the moment we blocked IL-6 receptor, we noticed that there was a decrease in time of bleeding in these mice. So these are orally infected mice with vehicle, treated with vehicle. You can see that they were bleeding a lot. And when we blocked IL-6 receptor, the bleeding time decreased next to normal levels, suggesting that IL-6 is indeed changing, it's compromising inflammation, is compromising the homeostatic cascade, and this can lead to this bleeding of infected mice. And then we went, and what happens with the patients? Is this real for the patients? We still don't know this question, but we do know that patients, as we showed, have alterations in out and eyes, as our mice does. So we did observe an increase in these hepatic enzymes in patients and in mice, suggesting that our model is going the same way. And we looked at 2 factors involved in the cascade of the hemostatic cascade. APC, which is a regulator of the cascade, and factor 7, which is involved in the activation of the coagulation cascade. And we can see that infected patients have changed in these 2 molecules, improved in APC, and look at factor 7, we can see clearly a number of patients that decrease the number of these factors in the blood, suggesting that these factors might be consuming and be activating the cascade of these patients. So I'm sorry for my English, it's not the best. I want to thank everybody that help us and works with us. So this is our team. This is the staffs of the lab. I am the coordinator of the group, but we have other people that works with us and help us a lot. we have now this team that is developing other studies involving coagulation and immune response in the oral cavity. And we have these students, Juliana Barreto de Albuquerque, Daniela Silva dos Santos, Valéria Regina, and Gina Antunes were the students, PhD students that in the time described this phenomenon that I showed you. And we have important collaborators, including our Ana Rosa Pérez that help us in the studies. And I want to thank the state of Pará, which is our partner for studies and development, educative development of initiatives. The LACEN, which is the Central Laboratory of Pará State, which help us to collect the samples and to understand these numbers of of persons infected by ear. And Senabio helped us with these bioluminescence studies that I showed you and our financial support.
Thank you very much, Juliana. We have questions. The first is there some study about the role of oral infection in canine Chagas?
No, I think it's a very interesting question because they are sentinels of parasites present in the region. So it would be very interesting to develop these studies. In fact, we are the main group working with that today. I wanted to sensibilize you because I think this is an important issue and people have to worry about this oral infection and don't believe that oral infection is just like any other infection. It seems quite different. We don't have it yet, but I believe that would be the same since we are talking about a mammal. Maybe it would be similar to the mice. It's important.
Okay. Thank you. You have other question from Rick Tarleton.
Mm-hmm.
Say, very nice talk. Could you elaborate on how you differentiate bacterial versus oral infection? Specifically, how do you know what some or many bacterial infections are not also via an oral route? It's an interesting question.
Yes, very interesting question. This was one of our subjects of work in the beginning. We have to differentiate vector routes by the oral routes. And it's basically analysis in patients. We look at signals of entrance in patients. So if the patient goes to the hospital and said, there was this bug, this triatomine, and they have entrance portans, lesion in the skin, it is classified as a vectorial route. But if the person goes to the doctor and said, I never touched that bug, I never saw that insect, he doesn't have entrance lesion, okay? And usually happens in an outbreak because people usually share the same food. You have one person that is not feeling well, goes to the doctor, and then they say this person has a positive diagnostic for Chagas disease, parasitological diagnostic. They start to talk. What were you doing 1 week ago when the symptoms started? Who is eating with you? Who is your family? They start to look and they can see that several people at the same family usually are infected as well. And they all doesn't have lesions or any signs of entrance of the parasites by the hematophage bug. So it's basically like that. This is an important question because some people said, so it might not be an oral infection. Can you prove that is an oral infection? Our way to prove is that they happen in outbreaks and they don't have this entrance lesion. And there is this study from this person in Amazonas. There's this doctor who worked and showed the acai was the fruit involved in this outbreak. He had a girl that was went to his office and said, I'm not feeling well, I'm not okay. And he did the diagnosis for oral Chagas disease. And he asked, what are you eating? What did you eat in the last days? And she said, a lot of acai. And I still have this acai in my house. And he said, whoa, bring me this acai, please. I want to see this acai. And when he got this acai, he found the parasite inside of the acai fruits. And she said, but I give this acai to all my family. And when they look at the family, a lot of them were infected at the same time. Sometimes were asymptomatic, sometimes were symptomatic, but the doctors didn't even think about that of oral infection. I don't know if I answer your question.
Yes, I think it's okay. Thank you very much. Here, there are others. Were the oral infection with tripomastigoid or metacyclis, and what dose? Do you think the infection site or course will differ in an animal ate bug versus ingested free parasite related with the food in people? This question is sorry, for Rick Tarleton too.
Okay. Well, we tested in the lab blood tripomastigoids and metacyclic tripomastigoids because we have a collaboration with, let me see here, Carlos José Moreira. They have insectarium, they sell insects, and we ask them to infect these insects and give us blood tripomastigoids and also metacyclic tripomastigoids from the insects. So we've developed it with both cases. In both cases, the infections were occurring similarly, okay? And we can also do that with culture parasites. We can also infect orally with culture parasites. There are some studies now suggesting that insects derived tripomastigoids, which means metacyclic tripomastigoids are more effective. And indeed, in some situations, it shows more effective than blood tripomastigoids. But I can tell you that we did it with culture, we did it with insect derived, and we have infection at the same way. What's the number of infection in our model? In our model, we tried the last we can. We found in mice, we used 50,000 tripomastigoids. But Hola. But what happens in nature, what happens in the field, we don't know. Okay, it's very difficult to think about a number that is necessary to infect people. One thing that we can hear the patient saying that, but Anna ate more acai than me and she's better. And I ate just a little bit of acai and I'm worse. So we don't know how to explain that. We think it's about immune response, but we don't know how much are these people eating able to infect. Some scientists that think about that, they say it's different in the vector routes. The the bug can release 100 parasites to go to the skin. And in the other situation, you are drinking a crushed vector with thousands of parasites. So this is a point of view. I don't believe that, because you have sometimes one bug in 1 liter of acai. So it's difficult to think that the person is drinking all the bug with all the parasites. So it's really hard to think about that. And I do believe that the root of infections change the behavior. Yes, I do. Our experiment shows if we infected mice intravenously, subcutaneously, orally, and intragashically, the pattern of infection will be completely different. We already saw that. The parasitemia is completely different, and survival is completely different. So I do think that if you have the same parasite in the same inoculi, if you change the pattern of infection, you have a completely different infection.
Thank you very much, Juliana. I have a question. You know that the only described case of oral infection in Argentina are from the late 60s for undercooking food. Do you think that could be the oral infection could be an undervaluated route of human infection in our country?
Anna, I can tell you about my experience in Pará State. That's why I brought you. It's not immunology, but I wanted to show you this experience in our state. I think so. There are some problems that we face with clinicians in the field, because they are usually trained to think like they were taught in the school. And you don't have oral transmission in our books of parasitology. You don't have this kind of talk in the university, in the medicine course. So it takes time to doctors, to clinicians, to sensibilize and to try to understand that. The symptoms are very specific. So you have fever, you have headache. So if you, depending on the region, for example, in Amazonia, fever, headache, it can be malaria, it can be a lot of things, dengue, it can be a lot of things, different things. So we have to keep talking to these clinicians. Please think about Chagas disease. You are in an endemic area, you have the vector, you have the parasite here. People are going to the forest, are getting their food, are cooking their food at home. They don't take sometimes the care they need to do that. So you must think about this possibility. And sometimes we can see in some cities that they don't have case because they don't even suspect of the infection. So they treat the patient with, they treat the fever, they treat the headache, they treat a lot of things, and the patients get chronic. Doesn't even know that he was infected already. And maybe the whole family is infected. So I would guess, this is a guess, I don't know, but in the Chaco, you have the vectors. The Chaco, you have the parasites. So I would be worried with the Chaco region. That was my guess.
Yes.
It's a guess.
Yes, it's really possible.
So if I was there, I was talking to the clinicians and think, please think about this possibility. If you have someone with fever, face edema, legs edema, please consider us to make a Chagas disease test, a parasitological test to consider Chagas disease. And sometimes in these outbreaks, you have only a few people that are symptomatic. So you have to catch these people who are symptomatic and to follow the other ones that are not. So in this case in Recife in 2019. There were more than 70 people infected at the same time. They were in a party. And only few people went to the right doctor who suspects Chagas disease. And then he knew about Chagas disease oral infection. He asked for the exams, and he thought, you are infected, you all are infected. And these groups that were in the party, they have a WhatsApp group. Today, technology is everything. And they said, people, we are infected with Chagas disease. Are you feeling well? And then 70 people were infected and didn't even know that they were infected. So I think sensibility is all.
Thank you very much, Juliana. We are on time. We need to pass to the next speaker.
Okay. So our next speaker will be Dr. Nuria Girones. Nuria is a professor in the microbiology area of the molecular biology. Of the Molecular Biology Department of the Universidad Autónoma de Madrid, and an independent researcher in the Molecular Biology Department of the Centro de Biología Molecular Severo Ochoa in Madrid. She has published more than 60 articles describing various aspects of the immunopathogenesis of Chagas disease, as well as the cardiac pathology and immunoregulatory responses promoted by Trypanosoma cruzi. So to end this first day, Nuria will tell us a seminar entitled Importance of the Immune Regulation in the Experimental Model of Trypanosoma cruzi Infection. Thank you, Nuria, and please share with us your presentation.
Okay, thank you, Andres. Thank you very much for inviting me to this symposium. And I would like to congratulate all the previous speakers, because it's been a very nice talk, excellent talks. And I will start the presentation now. As Andres told you, I'm going to talk about the immune regulation in the experimental model of Trypanosoma cruzi infection. And it's a work that we started many years ago, focusing in Chagas cardiomyopathy because of its high incidence. And because the mouse model, as everybody has already told, reproduces main features of the infection. The first thing that I want to introduce is the effect of the host and the dose of the parasites. This experiment, we show that non-susceptible mice survive to low and high doses of parasite of the Y strain, very effective one. And while BAP-C mice succumb to infection, die all by 21 days post-infection, and they show a small survival rate with low doses of infection. Also, the strains are important in the cardiac infiltration that we can see in the mice, from very mild to very strong with the Y strain, for instance. And this work has been done by former PhD student from Venezuela, the previous one, Cristina Sanoja, and the second one by Hector Rodriguez Angulo, who is in the audience right now. And so we started with this idea to try to characterize the lymphoid and myeloid cardiac infiltration in the mouse model. And for this, we use for some experiments both susceptible and non-susceptible mice, the Y strain, which is more reproducible for our studies. And at different days post-infection, we draw blood, elicit spleen, heart, and cells from bone marrow, and do different assays to try to characterize how the immune response is in these two different models. In particular, we do parasite load quantification by quantitative PCR, or RNA gene expression, protein expression, flow cytometry, some histology assays, and also we purify cells from the organs and try to characterize the protein expression and the capacity to proliferate, etc. So we started this work inspired about myeloid subsets in the cardiac infiltrate, inspired in some work by Susana Gea, who is also in the audience, I guess, and Pilar Aoki, in the Emmanuel Fresno's lab at CBMSO also, and with a PhD student, Enar Cuervo. And we were wondering whether the myeloid infiltrate in the heart could be either controlling parasite proliferation, killing the parasite through M1 macrophage proliferation, for instance, or whether we could have M2 macrophages which will promote parasite proliferation. And so we started to look at these markers of polarized macrophages as INOS, nitric oxide synthase, inducible nitric oxide synthase for M1 macrophages, or arginase for M2 macrophages. And the first experiments were impressive because bulk C mice, as you can see, arginase expression was huge, and also INOS expression indicating the presence of both types of responses in the mouse heart. And these enzymes were also present in no susceptible mice as black 6, but in a less amount. We were able to identify these enzymes in hair tissue by immunofluorescence, and we saw cells that expressed both. Expressed arginase, and they were macrophages because they expressed CD68. No other cells we could see that express this enzyme. And then we started purifying cells with magnetic beads from hair tissue, and so that at the first beginning we saw much more abundant expression of arginase 1 and INOS in cells CD11B purified from heart. They were more abundant also in bulk C than in black 6 mice, different days post-infection. And then we started wondering about other myeloid subsets that previously Manuel Fresno in the lab had characterized before, which were cells CD11B, GR1 positive, that were expanded during infection in the spleen. And they were, the cells were correlated with the expression of INOS, the the more inos they produce, the more inhibition of proliferation of other T cells in the cultures where was observed. And then we looked at in the literature how these myeloid suppressor cells through the years were better described, and we show this review from Gavrilovich, which is one of the people that more, the person that more studied these cells, and they are defined as derived from bone marrow, but instead of following the normal myeloid pathway of differentiation into macrophages, dendritic cells, or macrogranulocytes, they develop directly to an immature myeloid cell that can be found in pathologic conditions as tumor, trauma, or also in infections, although by the time that we started working with these cells, there were not many reports yet. How are these cells, MDCs? They express two surface markers. They can express the ones that are monocytic, express C11b, do not express LY6G, but do express LY6C, and they can be also granulocytic with expression of C11b and Ly6G, LY6G. The GR1 positive refers to an antibody that recognizes both LY6G and LY6C. So, for instance, in the previous slides, we were looking at both populations, monocytic and granulocytic, but later more antibodies were developed that can allow us to distinguish between these two monocytic or granulocytic MDCs. And besides these markers that we can differentiate between the two populations, there are several other markers, biomarkers, that can be used to characterize these cells. This was published by Bronte more recently, trying to find a better way to name these cells and to differentiate between them. And besides the expression of enzymes and these surface markers, they conclude that the definite proof of an MDC being a bona fide MDC is to demonstrate the suppressive function. Thus, we started trying again to purify these cells. We saw the phenotype using also magnetic beads. We were able to separate from CD11b positive the monocytic and the granulocytic, and we saw that in fact the ones that expressed arginase 1 inos were the monocytic ones, the ones that were LY6G negative and CD11b positive. These cells, besides looking at the expression, we were able to culture with spleen cells, and we saw that these monocytic NDCs were the ones that were able to inhibit T cell proliferation. And this inhibition was dependent on nitric oxide, because when we use nitric inose inhibitor, proliferation was restored. Also, we found the same kind of MDCs expansion in bone marrow. We saw that the number of CD11B through the course of acute infection decreased by day 10 post-infection, and despite this, the number of monocytic MDCs here in green increased by day 13, while did not show granulocytic cells, which decreased by the same day. Also, we found that these cells expressed inos and arginase 1, and they were able also to suppress the self-isolated from bone marrow monocytic MDCs were able to inhibit T-cell proliferation. The same we found in the spleen. In the spleen, we saw, contrary to bone marrow, a replenishment of the spleen, which causes splenomegalia, which is a characteristic of acute infection in mice also. And in particular, we saw this further expansion of myeloid monocytic MDCs by day 13. And in addition, in the spleen, we saw expansion of granulocytic MDCs. Again, the expression of the enzymes was confirmed in the spleen, and in addition, these cells were also able to inhibit T-cell proliferation. This work was in bone marrow and spleen, was done by Sofia Carvajosa, who also did her PhD in the lab. Moreover, it is known that arginase is able to metabolize L-arginine as well as inos does. They share this common substrate, and we wondered whether there was L-arginine extracellular depletion during infection, and this was the fact in non-susceptible black 6 mice. We saw from the normal basal levels of L-arginine, about 100 micromolar, we saw a great inhibition of decrease of L-arginine concentration by day 14, which was recovered in the case of the non-susceptible black 6 mice by day 21. In contrast, in BAP-C mice, we saw that the same similar depletion by day 14, but in addition, this was sustained through until day 21, and as we saw the survival curves, after that day, the mice died. So, apart from plasma, we also looked at L-arginine levels and other enzymes, metabolites that are involved in the L-arginine pathway, as ornithine, urea, proline, putrescine, citrulline, all products of L-arginine by the arginase route or by the inos route, which is metabolize the same amino acid. And we saw that there was also a decrease in heart of the L-arginine levels. And in contrast, there was an increase in asymmetric dimethylarginine, which is the ratio of L-arginine and ADMA. Decrease in this ratio is involved in some cardiac pathologies. So we wonder whether supplementing the mice with L-arginine could improve the outcome of the disease. And this is was done by Sofia Carvajosa. We can see here the parasitemia of the mice, BALB/C mice in this case, control in white, and the one supplemented with L-arginine in black. And we can see this double peak that was mentioned at the beginning of the session, and when how L-arginine is able to inhibit somehow this second peak of parasitemia. This was also had also effect in the survival of the mice, which once they were treated with L-arginine showed higher survival. And we started thinking how can be, which is the mechanism that allows this better outcome of the disease. And we used measured nitric oxide production in the control and the L-arginine treated mice, and saw that the levels of nitric L-arginine were restored first, and then plasma ADMA also remained, in this case, didn't change, but the levels of nitrates in plasma were increased in the ones that were treated with L-arginine. That is, the mice had this expansion of MDCs, which express arginase and inos, but because of depletion of L-arginine, nitric oxide production to block parasite proliferation was inhibited. So, on the other hand, we also studied some subsets of lymphoid cells in the cardiac infiltrate, mostly focused on CD4 cells, Th1, U2, Tregs, and Th17, involved in different outcomes of different processes, infectious processes, and immune alterations as autoimmunity. And this work was done by Cristina Sanoja, also doing her PhD in the lab. And we used an approach measuring mRNA expression of CD markers in cardiac tissue. And in red, you have the BALB/C, and in blue, the black 6. And you can see that despite BALB/C mice had much more parasite load in the heart, they had much lower expression of CD4 and CD8 than black 6, indicating that probably the cells infiltrating were more abundant in the black 6 model. Also, we looked at B cells, macrophages, and dendritic cells, and the immune response was much stronger in black 6 than in BALB/C. Also, we looked at different cytokines, and the same was Th1 cytokines, and we observed the same. And we comparing both bulb C and black 6 mice with looking at more cytokines. We saw that both types of cytokines were produced, induced, but the Th1, Th2 balance was always higher in black 6 mice compared to bulb C. And in particular, we looked at the markers of Tregs and Th17 cells, and saw that bulb C showed in the inflammatory infiltrate of the mice, cardiac infiltrate, they had a small proportion of IL-17, but no detected Treg cells, FOXP3. On the other hand, black 6 mice expressed a little, had a little proportion of regulatory T cells, but not Th17. And we looked at several cytokines and markers and matched with this observation. More regulation in black 6, in black, and more, for instance, IL-6 in bulb C mice. At the level of the plasma, also we saw that interferon gamma and IL-6 were the cytokines that were more abundant in plasma, and in particular, we saw that in bulb C, interferon gamma was similar between the bulb C and black 6 mice, but IL-6 was much higher in bulb C compared to black 6, both in using high inoculum and low inoculum of parasites. Those IL-6 was like a sign of a worst outcome of the disease, which is the case of bulb C. With all this, and to conclude, we draw this cartoon where in the top part we have susceptible mice that has a stronger Th2 cytokine profile, MDC expansion, which is related to increased levels of IL-6, which IL-6 is shown to inhibit regulatory T cells and to promote Th17 cells, and this leads to parasite proliferation and the death of the mice, together with this L-arginine ADMA ratio low that leads to the death of the mice. In contrast, the black 6, we have a stronger Th1 response, which is controlled by Treg, and less extent of Th2 response and MDC's expansion. And this balanced response leads to control of the infection and mice survival. And the question that we would like to ask is whether we can target these MDC cells. They are good because in small amounts, because it has been shown that by Susanna Hea that targeting, inhibiting these cells leads to death of the mice. But on the other hand, if we supplement with L-arginine, can promote survival also. So we believe that these cells are important in acute infection, And maybe related to the previous talk, in oral infection could be studied. And just to finish, all the people that has worked with these results, has contributed, and the persons that are now doing just finishing the PhD or doing it, and the technicians that collaborated with us, and all the collaborators, Susana Gea, Ricardo Corral, Felipe Bull, Hector Rodriguez, and all the funding and consortia that we participate. And thank you very much.
Okay, thank you, Nuria, very much for your talk. Really appreciate it. We have some questions. One from Gabriel Cabrera. He says, very nice talk. It is interesting that bulb C mice and black 6 mice have different susceptibility against different TCRC strains, such as Tulawen and Y strain. Since bulb C and black 6 have also different response in MDSS alterations, have you tried to analyze or correlate these alterations? Thank you. Your papers are very useful.
We think that the differences between Tulawen and Y strain in terms of MDCs may be related to the target organ that the parasites reach. We believe that Y strain is more related to cardiac pathology, and the Tulawen strain is more related to liver infection. And this can have different, the cells can have different outcomes in terms of survival. I don't know if I answered satisfactorily.
Okay. Now a question from Karina Gomez. Have you checked whether L-arginine treatment modified the myeloid cells phenotypes?
Not in detail. When we supplemented with L-arginine, we just looked at plasma levels of nitric oxide and L-arginine, but we didn't study the phenotypes. No, it's a good idea to follow this question.
Okay.
Thank you, doctor.
I have another one from Ana Rosa. If you have any preliminary result in humans that shows any relationship in between the levels of MDSC and the development or not of cardiac pathology?
We haven't not had a chance to look at MDCs in patients. It's more difficult in Spain to find good collaborations because and have people also to be ready to take the samples and do the work. But what we have seen is with some plasmas from Dr. Silvina Villar, we have seen that the levels with some plasma from Hector Rodriguez from Venezuela, we have seen some relation with the levels of L-arginine and the pathology. But still, I think these MDCs are more related to acute infection. And most of the samples that we get from either blood or plasma are from in Spain. All the patients are mostly indeterminate. We don't have much cardiac patients to compare. And I think it's a more acute effect that of MDCs, unless other can do more investigations and show the contrary.
Okay, and I have one from me. That is, if you had the chance to analyze the myeloid dendritic cells population in the spleen or lymph nodes in your experimental models.
No, we just look sometimes to CD11C marker expression, but we saw it was more expressed in black cyst mice, but we haven't had the chance to look further.
Okay, thank you very much. I think we are done with the questions. So we finally arrived to the end of our first day of Symposium. We would like to thank our great speakers again for their fantastic talks, and also the great audience that joined us today and was really from the beginning to the end. So thank you very much. Also, we would like to congratulate women in this special day for the distinguished work that you perform, not only in all the life cycle, but also in science. We'd like to bring you a little small piece of information. These talks and the following ones will be uploaded to the private YouTube channel of the University of Rosario anytime this week. So probably in the next Monday, we'll communicate the link to access in case you want to share this with your colleagues or to listen again to these wonderful talks. And finally, we hope to see you next Monday to continue learning immunology in Chagas disease. Thank you all for your participation.
Thank you.
Thank you. Hope to see you all next Monday. Bye-bye.
Bye-bye.
Bye-bye. Until the next
Thank you very much to all the organizers.
Thank you, Media.
See you next Monday.
The next Monday, yes.
Bye.
Okay,
Bye-bye.
Gracias a todos.
Adiós. Podés apagar el video, Ayo, si querés.